In this session, we're going to cover an overview of protein synthesis and highlight the differences between prokaryotic and eukaryotic organisms. So the basic principles in protein synthesis include transcription and translation, which are the two main processes that link a gene to a protein. Genes provide instructions for making specific proteins, and the bridge between DNA and protein synthesis is the nucleic acid called RNA. If you remember, RNA is chemically similar to DNA, except that it contains ribose as its sugar and substitutes the nitrogenous base uracil for thymine. In addition, an RNA molecule usually consists of a single strand. In DNA or RNA, the 4 nucleotide monomers act like the letters of the alphabet to communicate information. The specific sequence of hundreds to thousands of nucleotides in a gene carries the information for the primary structure of proteins, the linear order of the 20 possible amino acids. Getting from DNA to protein requires 2 major steps: transcription and translation. During transcription, a DNA strand provides a template for the synthesis of a complementary RNA strand. Just as a DNA strand provides a template for the synthesis of each new complementary strand during DNA replication, it provides a template as well for assembling a sequence of RNA nucleotides. Transcription of a protein coding gene produces a messenger RNA, also called mRNA molecule. Translation is the synthesis of a polypeptide using the information in mRNA. During translation, there is a change in language. The sites of translation are the ribosomes, complex particles that facilitate the orderly assembly of amino acids into polypeptide chains. Translation and transcription occur in all organisms from all 3 domains of life. In prokaryotes, translation of mRNA can begin before transcription has finished. In a eukaryotic cell, a nuclear membrane or nuclear envelope separates transcription from translation, as we'll see in these pictures. Eukaryotic RNA transcripts are modified through RNA processing to yield an mRNA. So if you look at this overview of the transcription and translation and the flow of genetic information, you can see that the basic mechanics of transcription and translation are similar in both eukaryotic organisms and prokaryotic organisms or bacteria. Because bacteria lack nuclei, their DNA is not segregated from ribosomes and other protein synthesizing equipment. This allows the coupling of transcription and translation. Ribosomes attach to the leading end of an mRNA molecule while transcription is still in progress. In a eukaryotic cell, transcription occurs in the nucleus and translation occurs at ribosomes in the cytoplasm. The transcription of a protein-coding eukaryotic gene results in a pre-mRNA. The initial RNA transcript of any gene is called a primary transcript, while further processing yields the finished mRNA. The finished mRNA then leaves the nucleus and goes into the cytoplasm, where the machinery for building the protein is found and translation occurs. To summarize, genes program protein synthesis via genetic messages in the form of messenger RNA. The molecular chain of command in a cell has a directional flow of genetic information: DNA, RNA, protein, sometimes referred to as the central dogma, which Francis Crick dubbed this concept in 1956. Although some RNA molecules can act as templates for DNA, this is a rare exception that does not invalidate the idea that, in general, genetic information flows from DNA to RNA to protein.